So repeats, they pose a great challenge to the assembly process. Why repeats are important? As we already know that eukaryotic genomes, and they are composed of major proportion of those eukaryotic organisms genomes is composed of repeats. And repeats, they can be a source of misassemblies. For example, if we look into this picture, we can have a normal sequence. So we have repeats. Here is R1 and the second copy of the repeat R2, and in between we have a normal sequence. So what happens if we take this part and put it here? So obviously since this part is repeated, so obviously the reads are put together and software might take that these two segments are the same thing, and then we might skip this part right here, and it can stay as an orphan contig. So repeat, if how can we solve this issue of repeat? So we can have some sequencing technologies. If we can get those read lengths which are longer than the lengths of the repeats, but unfortunately the technologies are not those long read producing technologies are not efficient. So we have to get back to those short reads. So what we can do next is we might use another option, that is we can use mate pairs. Right, so we have seen them earlier. So in this example, in this figure, what we see here is that we have the same part of the genome where we have those repeats, but here we are using those paired end reads. So how we get those paired end reads? We have seen it earlier in library preparations. What we do here is we can take those longer fragments and we can skip this middle part. We call it as insert, and then we can get those sequences from those ends. So in this way these ends are sequenced, but we know that what those pairs are, and for example this goes with this, we know that, this goes with this, we know that. So if we could keep track of these things, these reads, we can expect that these reads, they will align at a certain distance. So if they are not aligning at a certain distance, then obviously if they are close enough, that means we might have skipped some regions. So for example here, this read can be helpful while spanning this region, and these two reads, obviously this read which is there in this repeat and the second read which is there in this fragment which became orphan in the last example. So if we put them together, obviously if we are observing only this one in the final assembly and its partner is not there, say this is 3 and this is the bar, if that partner is missing, we suspect that we are actually missing some fragment. So in this way we can put this fragment inside by while keeping track of these paired end reads. So paired end reads help solving this issue of repeats. This process of generating those paired end reads is called as a double-barreled shotgun assembly. What we do here is, as we have seen quite a few times earlier, that we take those DNA fragments and then we sequence their ends, leaving a huge insert in between them. And what is the advantage? Obviously, it can, when we reconstruct assembly from those paired end reads, it can help solving the issues of repeats, because it is unlikely that both the members of the pairs are coming from repeats. So it is expected that one is coming from a repeat region and one may be coming from a normal region. So in this way we can put those repeats and normal regions in their perfect orders, as we see in this example. So here we have this normal sequence and then we have this repeat 1 and there is a repeat 2. So this is we have one assembly. So we can see the perfect orientation of these two folks, this one and this one. So in this way we can expect that there is another fragment, this one, in between these two repeats. So we can have, for example, here we have one of the paired end here in this normal fragment, the other one is in repeat 1, and then again in repeat 1 we have one of the pair here and the other one is here. So sometimes we do have misorientations, like for example this guy is here but its counterpart is here. So since this guy is kind of a repeat, we might neglect that. Same way we can have one fragment in R1 whose opposite fragment is here. So, but obviously the insert length is longer than expected, so we might kind of neglect this. So so far what we have seen is that most of the eukaryotic genomes they have repeats, and repeats they pose challenges to assembly, and using these double-barreled shotgun assembly methods in which we are using those paired end reads can help solve the issues of these repeats during assemblies.